Good morning, everyone. First, I would like to thank the Thai Immunology Society to invite me here to share my work. So in my lab, we study actually some broad. We study T cells. We also study the metabolism. We do a lot of metabolism in metabolic diseases. We also study microparticles, the extracellular vesicles. So we study the microparticles very early from the 2005, 2007. And we also study a lot of things about microbiology. So today I would like to talk about mechanical softness, paving the road of CAR-T therapy. Last 40 years, demonstration of the basic property of cells, mechanical and chemical signals together decide cell behaviors. So this field, microbiology, is emerging. 2020, I organized a lecture conference in Beijing. The title, Microbiology in Health and Disease. Actually, the sessions, beautiful sessions. For the organization, the 5 lecture, like the lecture biomedical engineering, lecture cell biology, lecture cancer, lecture immunology, lecture materials, all join this as the organizers. Unfortunately, due to the COVID-19, so finally this symposium canceled. So we still can find some information from the internet. We didn't do the conference in 2020. Last year, the similarly symposium was held in Boston, the March 19. So the title, Global Summit on Microbiology and Micromedicine in Boston. So I joined this meeting, this very summit, and a very small scale. 5 United States, 5 academic members joined this meeting. So the history of microbiology, the founder, Yuan Zhengfeng, just died in 100 years old, died in 2019. And the second generation leader, Xu Qian, both of them are the NAS, NAE, NAM, ANS members. So for this microbiology, that started in 1980s. They used the cell as a model to study mechanics. In 2000, early 2000, then the change. Use biomechanics to study cell behaviors. In 2020, they use mechanical learning to study diseases. So in my lab, around 2007, we use microbiology to study immunology and tumor. So what's the microbiology? What's the force? The principle in cells. ATP, hydrolysis mediated energy transition. What's mean? The ATP hydrolysis, this is potential energy. So this potential energy is released, transformed in the form of mechanical wave along microfilaments in our cells. The cytoskeleton, microtubules, intermediate filaments, and the third, microfilaments. So for the force, mechanical force, most mediated by the microfilaments, not by microtubules, not by intermediate filaments. So the mechanical action models, they have two types interaction, the force. The one type is cell extracellular matrix. The other is cell and cell. So to better understand the mechanical biology, so we need to know some terms like Young's modulus. Young's modulus is a measure of a solid material's stiffness or resistance to deformation. Stress equal to force divided by area, so the unit is Pascal or Pa. Young's modulus equal to stiffness. In our cells actually is materials, is totally materials. So the mechanical force in cells, tensile force divided by area equal to tensile stress. Cell resistance to deformation termed stiffness. Stiffness is reversed to softness. Softness equal to 1 divided by stiffness. So cell sense force of extracellular matrix, they use integrins. All the cells, the cell membrane, plasma membrane have the integrins. The integrins transduce mechanical signaling, also can transmit mechanical force. If we understand this, then we know cell have the stiffness, then tissue also have the stiffness. Cell stiffness should be matched with tissue stiffness. In our body, the most soft cell, unfertilized egg, this only 10 Pascal, 10 Pa. Embryonic stem cells also soft, 500 Pascal. Neuron nerve cells very soft. Our immune cells like neutrophils very soft. Macrophages, we see macrophages grows very big, very strong. We usually think macrophages very stiff. It's wrong. Macrophages very soft. Why? Softness equal to deformation. So macrophages are professional phagocytes. If the phagocytose extracellular component materials, they need deformation. This is why macrophages very soft. Neutrophils soft, macrophages soft. Differentiated tissue cells becoming stiff. So usually 1 to 5 kilo Pascal. In our body, muscle cells, skeletal muscle cells is stiffness, is the most stiff, 12 kilopascal. So for tissue, brain tissue, very soft, bone marrow, very soft, fat, very soft, muscle, stiff. The most stiff tissue is our teeth. Some stiffer than teeth is the culture dish. So in the in vitro experiment, all the cells culture in culture dish. It's artificial, never true in our body. So in my lab, we studied the mechanical biology. We use the mechanical principle to amplify the tumor stem cells, stem cell like the tumor cells. We call tumor repopulating cells, TRC. So this TRC will grow in the very soft fibrin gels. This gel only have mechanical signaling without chemical signaling. So we can amplify the TRC very, very well. So this is the soft TRC. We use a microfluid chip to sort the soft and the stiff cells. We found the softness actually is a basic property of stem cells. So how soft equal to stemness? We defined less than 300 Pascal is softness, is soft. So the cells with softness, most of the cells are stem cell-like cell types. So now we use softness as a physical marker to define the stemness. So how power of the softness? For example, how power of the tumor repopulating cells. So now we can use one TRC, one tumor repopulating cells to grow a tumor in wild type in immunocompetent mice to form a tumor. Even the differentiated tumor cells, partial differentiated tumor cells, in the soft gel, they can de-differentiation, only retro differentiation to become the stem cell-like tumor cells. Moreover, the tumor cells use the softness to counteract perforating pore formation. We know the CD8 T cells kill the tumor cells. They need the interaction at the immune synapse. At the immune synapse, the activated CD8 T cells release perforin. Then the perforin make a hole under the tumor cell surface, allowing granzyme entry into the cytoplasma of tumor cells. Then the granzyme B cleave the caspase 3, caspase 7 to induce tumor cell apoptosis. This is how the T cells kill tumor cells, the process. However, perforin making a hole, making a pore, this is a mechanical process. If the tumor cells are very soft, the perforin cannot making a pore very well. This means softness resistant to the T cell killing. However, we can use the tumor cell softness to kill tumor cells. We use a drug packaging microparticles. These particles are very large. The soft tumor cells use deformation to take up large size microparticles. However, these microparticles just deliver the drug from the lysosome to nucleus, then kill the tumor cells. So this is softness. Now we know the softness. Stem cells are very soft. Stem cells can be expanded in soft fibrin gel without differentiation. Softness induce retro or differentiation, only de-differentiation. Then we think, can we use softness to do something for T cells? So when we mention T cells, we sort of the naive T cells. We sort of effect T cells. We sort of memory T cells. effect T cells that have exhausted effect, pre-exhausted effect T cells. Memory T cells, central memory T cells, effect memory T cells. Now we know the tissue-resident memory T cells. Moreover, another concept, another type, they call stem cell-like T cells, which was identified in 2009. These stem cell-like T cells, TSC, make the tumor immunologists very excited. Why? Because the stem cell-like T cells survive very well in vivo. Also, stem cell-like T cells have very strong infiltration ability into the tumor site, into tumor mass. However, the shortage, the stem cell-like T cells, the number is very, very low in vivo. So far, we don't know the method to expand the stem-like T cells in vitro. Why hard expansion in vitro? This is involved in T cell fundamentality. We know T cells in Alzheimer's, positive selection, negative selection. After that, the T cells become naive T cells. All these T cells maturation, actually the TCR recognized self-antigen. So all the T cells in our body, they are potentially autoreactive T cells. They have the autoimmunity potential. So how our body control this? The TCR signaling, the TCR get activation through the TCR signaling. So the TCR signaling make the T cells proliferation. Also, they provide the program, make the T cell differentiation, make the T cell become exhaustion, make the T cell become senescence. So this differentiation, proliferation and differentiation, these two programs should be coupling. What's mean? If you choose proliferation, you lost stemless. If you choose stemless, you lost lumbar. So this is coupling. Lumbar and the stemless, you only choose one. So people try to make the stemless a lot of ways. For example, activating beta-catenin, inhibiting mTOR, inhibiting PSA kinase, all this inhibiting differentiation program. However, inhibiting differentiation program coupled with inhibiting proliferation. So what can we do? Can we use mechanical strategy, mechanical means for decoupling? We need to separate these two programs. So we just, for the CAR T cells, first you should activate the T cells. Conventionally, you put the T cells in the plate, in the plastic. However, we put the T cells in the fibrin gels, and then we add the viral vectors into the gel. So the viral vectors infect the T cells, then get the CAR T, the expression, the CAR. So we call this 3D CAR T cells. This mechanical-based CAR T cell expansion. So we can see this first day, they gel this one cells. 24 hours later, the group becomes several cells together. Day 2, day 3, and then 3 days, we draw the blood, the T cells from our blood. The 5 minutes, 3 days become 50 minutes. So that's a 10-fold increase amplification. Most important thing, the cells, all the cells, like the stem-like the CAR T cells. So for the stemless phenotype for T cells, they have the naive markers like a CD62 ligand, CD45RO negative. They have the CCR7 positive. They also have the memory phenotypes, TCF1, CD127 positive. They also have the effect phenotypes like CD195. So if you say phenotype, you need to do the self-renew assay in vitro. So we do the serial passage. We can, the 3D CAR T cells have this self-renewal, especially the stem cell markers. For T cells, people usually use the TCF1, but TCF1 actually is not a stemless marker. The real stemless marker, like the SOX2, LALOG, HEJOG, OCT34. We didn't find OCT34, but SOX2, LALOG, all upregulated. So this is truly self-renew. We also did the in vivo self-renew. After transfer, 30 days later, 1 month later, we can check this stem-like CAR T cells in blood, in bone marrow, in spleen. So the question is how mechanical signaling induces stemness of CAR T cells. To sense mechanical force, previously people demonstrated YAP is the sensor. However, YAP get activation sensing the collagen gel. For the fibrin gel, actually YAP get an inactivation. In fibrin gel, HAPO pathway get activation. So HAPO pathway mediate YAP degradation, the phosphorylate YAP. This phosphorylated YAP get degradation need an adapter. We identified the 1433 zeta. This protein adapter mediated the degradation. So the YAP suppresses MAFG expression. This is what we found. The MAFG is a big family. The biggest is the AP1. They have large MAF, small MAF. For this family, is a transcription factor. They have the basic region to binding DNA. They have the leucine zipper for the protein-protein interaction, protein-protein binding. So the MAFG is a small MAF, is a heterodimer transcription factor that just directly promote stemless gene expression. So how fibrin gel activate the adapter 1433 zeta? The chemical signaling, because the sensor uses the integrin, we found the chemical signaling can activate integrin, but integrin beta 2 without phosphorylation. However, mechanical signaling can phosphorylate the integrin beta 2. If we mutate this serine site, the loss of function. So now it's clear. The fibrin gel, the mechanical signaling, make the integrin beta 2 phosphorylation. The 14-33 protein, adapted protein, that recognize two phosphorylation proteins. One is integrin beta 2, another is phosphorylated YAP. Then mediate YAP degradation to the through the ubiquitination pathway. Another question, how fibrin gel promotes CAR T cell proliferation. YAP degradation, this make stemless, but the T cells, they get the proliferation in the gel. So how the T cells get the proliferation? The integrin beta 2, phosphorylated integrin beta 2, interact with the FAK, focal adhesion kinase. This kinase activate the ERK, the MAP kinase ERK. So the ERK promotes T cell proliferation. Now finally, what's the difference between chemical and mechanical? What's the difference? The chemical actually is receptor ligand interaction, is antigen antibody interaction. This interaction, they use the weak bond, like the hydrogen bond, ionic bond, van der Waals forces. Mechanical force, they use ATP hydrolysis, release the energy. So this energy magnitude is much larger than the weak bond. Also, the mechanical force have the orientation, also have the frequency, So all this totally different from chemical signaling, So now we understanding how the CAR T cells get expansion in the fibrin gel in the soft mechanical microenvironment. So these CAR T cells have the stemless properties. So can we use such CAR T cells for cancer treatment? CAR T cells for blood malignancy, the treatment outcome is good. But CAR T cells for solid tumors, no efficiency. Efficacy is bad. So we try to use the CAR T cells to solve the solid tumor treatment. So first we do the in vitro serial killing. We can say they have a long-term killing capacity. How about in vivo? So we use a human colorectal cancer model. We can say they enhance the CAR T cell infiltration and the survival in tumor mass. As a result, the tumor growth inhibited and the mice prolonged survival. We also use the human glioblastoma. We also tested pancreatic cancer. All the results similar. So we can do the comparison in rigid plate, the culture dish, the plastic, the integrin beta 2 not getting activated without phosphorylation. So the YAP ranks to heterodimer. In the nucleus, the block MAFG. So the cell get a differentiation. However, in the 3D fibrin gels, the mechanical force, the phosphorylated integrin beta 2. This phosphorylated integrin beta 2 activate FARC and ERK to make the cell T cell proliferation. At the same time, recruit the 14338 adapter protein. This adapter protein mediate YAP degradation. YAP degradation. So the MAFG released. Then the MAFG trans-activate stemless genes to confer the amplified T cells with stem-like properties. So the softness, the mechanical softness, probably is the next generation CAR T therapy. For this softness generated CAR T cells, they have very strong infiltration, can survive in tumor mass, so they can kill the tumor cells. Meantime, these softness T cells, we use very short time for expansion, only 3 to 5 days. And also we didn't add any growth factor without cytokines. So very, very cheap. Very, very cheap. So this is why we do research. We want to use this for patients. So finally, I would like to say this idea actually in older time, for example, in older Chinese philosophy, they call the Tao Te Ching, the writing Lao Tzu. So the softest thing in the world dashes against and overcomes the hardest. Everyone in the world knows that the soft overcomes the hard, and the weak the strong, but no one is able to carry it out in practice. Now we just borrow a little for our research. So finally, I would like to thank all our lab members, and also thank the grants from our institute that comes, and also from the National Science Foundation for the basic science center program. Thank you.